The title of this presentation tonight is How Ribosomes Point to Design. Ribosomes are tiny assemblies of proteins and RNA found in the cells of all organisms, whether animals, plants, algae, or bacteria. I will cover what ribosomes are and what they do. I will compare ribosomes to a man-made machine, and I'll use something called the watchmaker argument to show that ribosomes must have been designed. And then I'm going to tell the stories of the two COVID-19 vaccines, which are based on messenger RNA. Ribosomes manufacture all the proteins in your body. That is to say, they are tiny little machines which take a pattern from your DNA and use that pattern to build every protein that your body needs. During the balance of this presentation, I will show how they work, and I'll use something called the watchmaker argument to claim that they are so similar to a common machine, which was obviously designed and built by intelligent beings, that it is reasonable to infer that they also were intelligently designed. The human body contains approximately 20 million trillion ribosomes in round numbers. Here's another look at that number. It's the number 20 followed by 18 zeros, or about 1/2 million ribosomes for every cell in your body, although many of your cells have a lot more. So they must not be very big, right? Right. A ribosome is about 23 nanometers across. A human hair, by comparison, averages about 75 microns across, and there are 1,000 nanometers in a micron, so you could stack 3,300 ribosomes side by side in the width, not the length, of a single human hair. Here's a picture of a typical animal cell. The purple structure in the upper right, that is this thing here, is the nucleus where DNA resides. If you look closely, you can see some tiny ribosomes on the surface of the nucleus, not the little yellow things, but the really tiny things on the surface of the nucleus and on surfaces of other parts of the cell here. They are the tiny purple dots, and they're not these secretions that are exiting the cell in the lower right. We'll come back to this picture in a moment. Here's a picture of DNA in its helical coil structure. DNA is found in the nucleus of the cell. In the upper right corner is a chromosome. DNA is tightly wound around proteins in the chromosome, and in this picture, the DNA is unwound to reveal a gene. This gene consists of 2 exons and an intron. In making a protein, any introns are cut out of the gene and only the exons are translated into the protein. Here's a close-up cartoon of some DNA. DNA is made up of the so-called nitrogenous bases adenine, or A, thymine, T, cytosine, C, and guanine, G, in a complementary double-stranded helical coil where A is always matched up with T and C is matched up with G, and vice versa. The ribosome actually uses RNA, not DNA, so here's a cartoon of what RNA looks like alongside DNA. Note that RNA has the same nucleotides, adenine, cytosine, and guanine. However, uracil is substituted for thymine. To make a protein, DNA is first transcribed into RNA, and the introns are cut out of the RNA strand, which is now known as messenger RNA, or mRNA. The mRNA exits the nucleus and finds a ribosome. Most of my argument for design in the ribosome is focused on the translation process, which I'll describe in the next few slides, but the transcription process also shows design in at least 2 areas. Transcription is carried out largely by complex proteins called RNA polymerases that attach themselves to the DNA strand, separate the 2 strands, and copy the DNA from one of the strands onto a new strand of RNA. copying the adenine, cytosine, and guanine is pretty straightforward, but how and why does thymine get converted into uracil? Turns out that uracil is a better choice for the process, but if an evolutionary process is undirected and has no goal, how could it make such a choice? Once the RNA strand is copied, the next step is to cut out any introns on the strand. Again, how could an undirected process make such a choice? Here's our picture of the typical animal cell again. The little yellow daisy-shaped structures which we overlooked before on the surface of the nucleus are nuclear pores through which the mRNA exits the nucleus. Note, just outside the nucleus is a blue folded structure, this thing here, called the rough endoplasmic reticulum. And this is where most of the ribosomes are located. And if you look sharply, you can actually see them on the rough endoplasmic reticulum. The mRNA which came from the gene is about to be translated into a protein by the ribosome. The mRNA is subdivided into groups of 3 nucleotides called codons. This is the genetic code which shows how this translation process occurs. There are a total of 64 codons ranging from UUU at the top left to GGG at the bottom right. The ribosome translates the codons into amino acids represented here in blue. So UUU, again at the top, at the upper left, translates the amino acid phenylalanine, or PHE, this one here. The code contains some redundancy, which reduces errors in the translation process. So phenylalanine, in addition to being translated by UUU, is also translated by UUC. It turns out that there are many other codes with similar redundancy, which life could have used, but this one is actually optimized for correcting translation errors. Note that one of the amino acids, methionine, shown here in green, is shown in green, not blue, because most proteins actually start with methionine. So the codon AUG is often called the start codon. Notice also that 3 codons, these ones up here labeled stop signals, or stop codons, are stop signals, and they're represented in red. UAA, UAG, and UGA. When they're encountered by the ribosome, they signal an end to the protein chain. As I'm about to claim, this is an exact match to the code used by the man-made machine that I'm comparing to the ribosome. Here's a list of all 20 amino acids found in every living organism. Excuse me. The pink bands in the center of the 2 columns, here and here, mark the structure that is common to each of the amino acids. The R groups, also known as functional groups, are shown in the middle, and they're what makes each of the amino acids unique. So in the upper left-hand corner, alanine, represented ALA in the genetic code, has a CH3 here, or methyl R group. Again, this eye chart is far more detailed than you need for this presentation. At this point, you should just be aware that there are 20 amino acids, that in the genetic code they are abbreviated by a 3-letter code, and that each one is translated by 1 or more 3-letter codons in the genetic code. Ribosomes again build the proteins in every living organism, from the tiniest bacterial cell to the largest animals, to every plant and tree and flower. Ribosomes are actually made of a combination of proteins and ribosomal RNA, or rRNA. Notice a slight paradox. If ribosomes are made of proteins and also build the proteins, which came first, the ribosome or the protein? Well, let's ignore that paradox for the moment. Bacterial ribosomes are somewhat smaller than those in more complex organisms like animals and plants. Each ribosome consists of 2 pieces, a large subunit and a small subunit. the large and small subunits of the ribosome assemble around a strand of messenger RNA as the protein chain begins to be assembled. Here's a cartoon of the large or 50S and small or 30S subunits. The middle loop, known as the P site, this thing here, of the 3 loops in the large subunit is where the translation actually occurs. The messenger RNA enters the ribosome here between the large and small subunit, where the ribosomal RNA aligns the messenger RNA properly. The next structure we need to look at is the transfer RNA, or tRNA. The diagram on the left shows that tRNA is actually a length of RNA which folds up into a 3-dimensional structure, kind of looking like the picture that you see there. The diagram on the right is a cartoon of the same thing without all the busyness of the RNA. Remember that the genetic code shows codons, but tRNA actually uses anticodons. This is because the anticodon links up with the codon. Remember that A always links with U and G always links with C. So if the messenger RNA in this case carries the codon UUC, this tRNA will link up with it, the anticodon being AAG, and the corresponding amino acid will be attached at the top of the tRNA, and it will be phenylalanine, or PHE. If that's confusing, hopefully this short video clip will help. It shows how the messenger RNA is translated into an amino acid sequence, which then becomes our protein. So now let's look at the video.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
A key part of the translation process is transfer RNA. Here's a model of a transfer RNA. This one codes for the amino acid phenylalanine, which is the same amino acid we looked at before the video, but with a different cartoon representation. At the bottom on the right-hand side, you can see 3 small bars pointing up and to the right. That's these 3 little green bars here. And these comprise the anticodon that will match up with the codon at the ribosome when the messenger RNA arrives. At the top right, up here, is the site where the amino acid will be attached. There are a specific set of 20 molecules which attach each amino acid to the appropriate transfer RNA matching the anticodon. These molecules are called aminoacyl tRNA synthetases, and we'll have more to say about them in a moment. The next set of slides represents text and pictures from a website called Molecule of the Month, a joint effort of several universities and US government agencies. I'm using these basically as is because they're well written, pretty easy to understand for the layperson, and strongly, I think, suggest design, but are written from a materialist evolutionary viewpoint. Since the process of DNA-directed protein synthesis was discovered, scientists and philosophers have searched, more or less seriously, for a relationship between the triplet nucleic acid codons and the chemical nature of the amino acid. These attempts have been uniformly unsuccessful, but remain an occasional topic of speculation because of their possible insights into the origins of life. There does not appear to be a specific interaction between the codons and the amino acids themselves. Instead, the match is made by transfer RNA, the Rosetta Stone that translates the nucleotide language of codons into the amino acid language of proteins. This translation is physical and direct. At one end of each tRNA is an anticodon that recognizes the genetic code, and at the other end is the appropriate amino acid for that code. And here's our phenylalanine tRNA again. Errors in the production of proteins can occur at both ends of the tRNA. The proper amino acid must be added to the tip of the tRNA, ready to be added to a growing protein chain. A battery of enzymes termed aminoacyl tRNA synthetases are in charge of this job. They generally make a mistake in about 1 out of every 10,000 tRNA molecules that they charge with an amino acid. Errors may also occur at the other end of the tRNA when the anticodon matches up with a codon. It may simply match improperly. This happens about 1 time in 500. Or, because each codon is 3 nucleotides long, it may associate in a shifted position instead of the proper reading frame. This will throw off the rest of the protein as each successive tRNA lines up in the wrong position after the shifted one. Fortunately, when genetic sequences are read in the wrong frame, they are filled with stop codons, so the protein synthesis will be aborted after a few dozen more amino acids are added. Transfer RNA molecules are composed of one short chain of RNA, 70 to 90 nucleotides in length, folded into a trefoil or clover shape. 2 different molecules are shown here, phenylalanine once again, and yet another cartoon representation, and aspartate tRNA. The 2 ends of the RNA chain are close to one another at the pointed end of the L-shaped structure at the top in this illustration. The amino acid is added here at the position shown by the arrows. The center of the chain forms the rounded leg of the L, shown here at the bottom, exposing the 3 nucleotides that form the anticodon. The other 2 loops of the trefoil are bundled into the elbow, where they provide structure to the whole molecule. In order to pair the right amino acid with the right anticodon, another set of proteins is used. They are called aminoacyl tRNA synthetases. When a ribosome pairs a CGC tRNA with a GCG codon, it expects to find an alanine amino acid carried by the tRNA. It has no way of checking. Each tRNA is matched with its amino acid long before it reaches the ribosome. The match is made by a collection of remarkable enzymes, again called the aminoacyl tRNA synthetases. These enzymes charge each tRNA with the proper amino acid, thus allowing each tRNA to make the proper translation from the genetic code of DNA into the amino acid code of proteins. Most cells make 20 different aminoacyl tRNA synthetases, one for each type of amino acid. These 20 enzymes are widely different, each optimized for function with its own particular amino acid and the set of tRNA molecules appropriate to that amino acid. As you might expect, many of these enzymes recognize their tRNA molecules using the anticodon, but this may not be possible in some cases. Take serine, for instance. 6 different codons specify the amino acid serine, so seryl tRNA synthetase must recognize 6 tRNA molecules with 6 different anticodons, including AGA and GCU, which are entirely different from one another. So tRNA molecules are also recognized using segments on the acceptor end and bases elsewhere in the molecule. Note also that each enzyme must recognize its own tRNA molecules, but at the same time, it must not bind to any of the other ones. So each tRNA has a set of positive interactions that match up the proper tRNA with the proper enzyme, and a set of negative interactions that block binding of improper pairs. Notice the implication here. Even though there are only 20 amino-acyl tRNA synthetases, there must be 64 different tRNAs corresponding to the 64 different codons in the genetic table. In this picture, 5 complexes, again, these are cartoon representations. They're not actual pictures of the tRNAs and the amino-acyl tRNA synthetases. But they do show the amino-acyl tRNA synthetase with tRNA aligned so that the tRNA molecules, which are shown in pink here, are in the same orientation. Notice that the enzymes approach the tRNA from different angles. So we have here isoleucine in the upper left-hand corner, valine in the upper right-hand corner, and glutamine in the middle right. And these enzymes cradle the tRNA, gripping the anticodon loop at the bottom in each tRNA, and placing the amino acid acceptor end of the tRNA in the active site at the top right in each tRNA. These all share a similar protein framework known as type 1, these 3, that is isoleucine, valine, and glutamine. and they all approach the tRNA similarly and add the amino acid to the last 2' hydroxyl group in the tRNA. The phenylalanine and threonine enzymes at the bottom left and bottom right are part of a second class of enzymes known as type 2. They approach the tRNA from the other side and add the amino acid to the other free hydroxyl on the last tRNA base. Amino-acyl tRNA synthetases must perform their tasks with high accuracy. Every mistake they make will result in a misplaced amino acid when new proteins are constructed. These enzymes make about 1 mistake in 10,000. For most amino acids, this level of accuracy is not too difficult to achieve. Most of the amino acids are quite different from one another, and as mentioned before, many parts of the different tRNA are used for accurate recognition. But in a few cases, it is difficult to choose just the right amino acids, and these enzymes must resort to special techniques. Isoleucine is a particularly difficult example. It is recognized by an isoleucine-shaped hole in the enzyme called the catalytic site, which is too small to fit larger amino acids like methionine and phenylalanine, and it's too hydrophobic or water-hating to bind anything with polar side chains. But the slightly smaller amino acid valine, different by only a single methyl group, also fits nicely into this pocket, the catalytic site, binding instead of isoleucine in about 1 in 150 times. This is far too many errors, so it doesn't really happen this way. Corrective steps must be taken. Isoleucyl tRNA synthetase solves this problem with a second active site called the editing site, which performs an editing reaction. Isoleucine does not fit into this site, but errant valine does. The mistake is then cleaved away, leaving the tRNA ready for a properly placed isoleucine amino acid. And this proofreading step improves the overall error rate for this tRNA to about 1 in 3,000. These enzymes are not gentle with tRNA molecules. This structure of glutaminyl tRNA synthetase with its tRNA is a good example. Here the tRNA is the upside down L-shaped structure of red, gray, and purple, and the synthetase enzyme is the green structure on the right. The enzyme firmly grips the anticodon down here at the bottom, spreading the 3 bases widely apart for better recognition. At the other end, up here, the enzyme unpairs 1 base at the beginning of the chain, seen curving upwards here, and kinks the long acceptor end of the chain into a tight hairpin, seen curving downward here. This places the 2' hydroxyl on the last nucleotide in the active site, where ATP and the amino acid not present in the structure are bound. Although I haven't mentioned it earlier, 1 adenine triphosphate molecule, or ATP, which is the important energy molecule of the cell, is present in each of the amino acyl tRNA synthetase molecules. And now let's watch a short video to put this all together.
Amino-acyl tRNA synthetases catalyze the formation of charged transfer RNA. To begin the process, a specific amino-acyl tRNA synthetase binds a particular amino acid and also a molecule of ATP at its active site. A covalent bond is formed between AMP and the carboxyl group of the amino acid, and pyrophosphate is released from the enzyme. A specific tRNA that has an anticodon that corresponds to the amino acid then binds to the synthetase. The RNA sequence in the anticodon region, as well as other parts of the tRNA molecule, such as the acceptor stem, are important for recognition between the tRNA and the amino-acyl tRNA synthetase. The bond is broken between the amino acid and AMP, and the AMP is released. At the same time, a covalent bond is formed between the amino acid and the 3' end of the tRNA, which is now called a charged tRNA molecule. The charged tRNA is then released from the enzyme and is now ready to be used in the process of translation on a ribosome.
Before we go on, I want to remind you of what we've just seen. First, DNA is transcribed into messenger RNA in the nucleus. The messenger RNA is transported to the ribosome through those nuclear pores we saw before. Second, transfer RNA is charged with the appropriate amino acid. Third, in the ribosome, the anticodon on the transfer RNA matches up with the codon on the messenger RNA. The amino acid on the transfer RNA is added to the polypeptide sequence being formed. The polypeptide is released from the ribosome when a stop codon is encountered. And finally, the proteins have to fold to be useful. How this folding happens is not straightforward, but it is beyond the scope of this lecture. Finally, just a reminder that the molecules we've encountered, besides the ribosomes and messenger RNA, include 64 different tRNAs before being charged with the proper amino acid, another 64 tRNAs after the charging so that they do contain the correct amino acid, 20 different amino-acyl tRNA synthetases, 20 different amino acids, plus a molecule of ATP for each tRNA, all at every ribosome in multiple copies, and which have to work efficiently together without shortages or surpluses. I think this is pretty amazing. It suggests to me that there must be some sort of coordinating process which is going on to keep things running smoothly. However, I'm not aware that any process like this has been proposed or observed or even suggested. At the end of this talk, we'll see that the 2 leading producers of the COVID-19 vaccines have made use of this process to manufacture their vaccines. Many of you may remember the basics of the watchmaker argument, which I've talked about in previous talks, but here's a review. In 1802, the Reverend William Paley published this book entitled Natural Theology, and in it he put forth this argument. You stumble on a watch in a field. You realize it has not always been there, in contrast with a rock, which very well may have been there forever. You see that the watch is intricately designed and elegantly made, you conclude, by an intelligent being. You realize that living creatures also appear intricately designed and elegantly made, and further, they have the ability to reproduce themselves, a property which even the most beautifully constructed and finest watch does not possess. It is reasonable to conclude then that living creatures are also designed by an intelligent being. Although it was possible even as early as 1802 to see elegance in the form of living creatures, not a lot of details were available, especially at the microscopic level. Many scientists dismissed Paley's argument because they believed humanly designed mechanisms were very different from living creatures. Today, however, biological science has progressed to the point where it is possible to make detailed comparisons between the parts that make up living creatures and humanly designed mechanisms, which gives new legs to the watchmaker argument. Here's a picture of a teletypewriter. These were ubiquitous in newsrooms of radio and TV stations, newspapers, and in military installations before the widespread use of computers and digital communications. These machines reached speeds of 100 words a minute as early as the mid-1940s, and they eventually got up to 300 words per minute. They could both send and receive messages, but it's the receive function which I argue closely resembles the function of the ribosome. Basically, a written message was translated into a string of binary characters, that is, ones and zeros, using a special code called ASCII, which stands for American Standard Code for Information Interchange. The message was then transmitted either over a telephone wire or a radio circuit. It was received by a machine like this, and the machine translated the ones and zeros back into letters and numbers using the ASCII code. The machine printed the message exactly as it was written. Before machines like this were available, an earlier machine worked in a similar manner, but the message was printed out on sticky tape, which was pasted onto yellow paper in the form of a telegram. The ASCII code has 128 7-bit bytes, similar to the 64 codons of the genetic code. Thus, ASCII is a binary or base 2 code, which uses bytes that are 7 bits long. What is actually transmitted over the telephone wire or circuit is a series of binary pulses or bits created simply by current being turned on or off. The letter capital A, for instance, would be 1000001, and small a would be 1100001. Recall that the genetic code uses 4 characters, the letters A, C, G, and U, which makes the genetic code a quaternary or base 4 code. A quaternary code can be converted into a binary code simply by using 2 bits for each of the letters A, C, G, and U. Thus, A could be represented by the bits 00, C by 01, G by 10, and U by 11. Notice that the ASCII code has start and stop signals like the genetic code. This is because a teletypewriter machine must work at exactly the same speed as the signal it is receiving, and the start and stop signals make syncing the signal and the machine easier. Here's an artist's depiction of a ribosome and a photo of a teletypewriter. Ribosomes are microscopic molecular structures that build proteins from DNA templates called genes. Every organism on Earth, from the tiniest bacteria to the tallest tree to the largest animal, contains a whole lot of ribosomes, and these ribosomes construct all the proteins in these organisms. In a single bacterial cell, there may be tens of thousands of ribosomes and up to 10 million in a mammal cell. the actual number is variable and increases dramatically as cell division occurs. A teletypewriter is pretty much like a large typewriter. You could sit at the keyboard and type out a message just like a typewriter, but you are also creating a punched paper tape of the message, which could be used to send your message out over radio waves or copper wires. The teletypewriter could also receive and decode these radio signals. So how could I possibly compare a ribosome and a teletypewriter? They clearly don't look anything alike, and they're vastly different in size. Functionally, however, they perform similar tasks. And here's a table showing some of their similarities. First, both are governed by codes. Second, they each take a message in a serial data form and convert it into another message in serial data form. RNA codons for ribosomes and ASCII bytes for teletypewriters. Third, both translate an input signal into something new. Ribosomes translate RNA codons into proteins. Teletypewriters translate ASCII code into English text. And finally, both have a serial output. Ribosomes produce proteins, not just amino acids, but proteins in a string, and then folded, and teletypewriters produce messages. Obviously, besides the vast size differential, there are also many differences between ribosomes and teletypewriters. Most of these differences are merely cosmetic. I will only mention the two that I consider most significant. One, amino acids may be represented by more than one codon. For instance, the amino acid serine is translated by the codons UCU, UCC, UCA, UCG, AGU, and AGC. The stop function by UAA, UAG, and UGA. This is functionally important because it gives the genetic code a form of redundancy, which provides a certain amount of error tolerance. The ASCII code has no redundancy. Each character represents one and only one letter, number, or function. Also, the ribosome generates the very proteins that make it up. That is, it is self-replicating. The point is that the ribosome is actually more complex than the teletypewriter. So, some conclusions. We have seen that ribosomes and teletypewriters have a lot of similarity, and both are very complex. Number two, we've also seen that the most significant differences between the ribosomes and teletypewriters suggest that ribosomes are more complex than teletypewriters. Third, teletypewriters were obviously designed by intelligent human designers. Fourth, if teletypewriters were designed and ribosomes appear to be even more complex than teletypewriters, it is reasonable to conclude that ribosomes were also designed. And finally, using the watchmaker argument, this suggests a designer. Maybe you're now thinking all of this is pretty obviously signs of design, but the majority of evolutionary biologists don't see it that way, including many who are Christians. A key aspect of the scientific method is the concept of methodological naturalism, which attempts to explain and test scientific endeavors, hypotheses, and events with only natural causes and events. Supernatural causes are not permitted. This means that another explanation must be sought for any process that looks designed. So many evolutionary biologists will look at the facts I've just presented and attribute them to evolution, not to design, even though they may not have ready explanations for how evolution could have produced what we see in nature. Design is not an acceptable answer, no matter how compelling the evidence may be. Many religious scientists say that their religious beliefs affect the way they think about the implications, often moral, of their work, but not the way they practice science. To say, God must have done it, is to abandon the scientific method. Methodological naturalism contrasts with metaphysical naturalism, a philosophic worldview and belief system that holds there is nothing but natural elements, principles, and relations of the kind studied by the natural sciences. This would be the worldview of atheists. This idea became popular during the Enlightenment in 17th and 18th century Europe, as some philosophers and scientists came to reject the idea that God was in control of the universe. Before I go on, I want to briefly suggest that there are many aspects of the processes I've just described that I don't have answers for, and I've not been able to find any in the scientific literature. It's possible that I've overlooked it, but I have at least spent some time thinking about some of the questions and looking in vain for scientifically acceptable answers. Here are just two of the questions which puzzle me. First, surrounding each ribosome, and there's a lot of ribosomes in every organism, but surrounding each ribosome must be 64 tRNAs corresponding to the 64 codons in the genetic code and carrying 20 different amino acids. How can the right match be made between the right mRNA codon and the right tRNA anticodon in an efficient manner? And what actually propels the tRNAs to their destination? Is it just chance? We know that and number two, we know that the majority of proteins actually contain metal ions, such as iron, magnesium, manganese, and copper, and lots of others. But these ions aren't part of any of the amino acids. How do they actually get incorporated into the proteins? I'm not suggesting there are only supernatural answers to these questions. In fact, I suspect that there are, in fact, naturalistic answers to them. And I believe if and when those answers are discovered, they will actually strengthen the case for design and further complicate the case for an evolutionary explanation. This has all been kind of theoretical and maybe a bit hard to imagine, but now I'd like to get a little bit more down to earth. Here's a picture of a COVID-19 virus particle. The virus genome was sequenced early last year and contains 29,811 base pairs. Viruses consist basically of RNA and proteins. Viruses work quickly when they enter our bodies. They attach themselves to a cell, invade the cell wall, and insert their RNA into the cell where it incorporates itself into the cell's DNA. The cell then replicates multiple copies of the virus, which proceed to destroy the cell and spread through the body. This is how we become infected by the COVID virus, as well as many other viruses, such as cold and flu. One of the amazing properties of our immune systems is that when they encounter a particle or substance they don't recognize, they immediately go into overdrive trying to reject it from our bodies. But it takes longer for this to happen than for the virus to replicate and spread. So the idea for a good vaccine is to introduce a particle that is similar to a part of the virus without being infectious. So when the immune system encounters that particle on the real virus, it is able to fight it off and the virus as well before infection sets in. Obviously, you don't want to introduce enough of the virus in the vaccine so that the virus could spread in the body. There are currently several techniques which have been used to develop COVID-19 vaccinations. One of the most successful so far uses a small piece of synthetic or man-made messenger RNA, which codes for a small protein on the COVID-19 spikes. That's the red things that you see here. If and when you get a COVID-19 or COVID vaccination shot, it will likely be either from Pfizer and its partner BioNTech or Moderna, both of which use the messenger RNA technique. Let's unpack that a little. Recall that to make a protein in the human body, DNA is first transcribed into messenger RNA in the cell's nucleus. Then the messenger RNA exits the nucleus through one of the daisy-like nuclear pores and finds a ribosome. The messenger RNA then threads its way through the ribosome, one codon or 3 nucleotides at a time, where each codon is then translated by transfer RNA into one of the 20 amino acids and the amino acids are strung together in a single strand called a polypeptide, which then undergoes folding to become the new protein. Creating a vaccine from mRNA skips the transcription step, starts with a piece of synthetic messenger RNA. This messenger RNA is then translated into the new protein by transfer RNAs in a ribosome. Since this is a protein from the COVID virus, the immune system won't recognize it and will attempt to build up an immunity to it. This takes time. Both the Pfizer and BioNTech and Moderna vaccines require 2 vaccinations, 3 weeks apart, and within 3 to 4 months have been shown by clinical testing to be 95% effective against COVID-19 infections. The story of how messenger RNA came to be used in vaccines reads almost like science fiction, and some of the participants will very likely be nominated for Nobel Prizes for their efforts in making it happen. Here's the story. This is Katalin Karikó. Her story is one of multiple setbacks before ultimate triumph. She earned a PhD in biochemistry from the University of Szeged in Hungary. She was fired from the Szeged Biological Research Center in 1985. I don't know why. She came to the US, where she became a professor at the University of Pennsylvania Medical School. By 1990, she had decided to try to harness the power of synthetic or man-made mRNA to fight disease. But the idea was too far-fetched to get enough funding to pursue. Although researchers at the University of Wisconsin had made it work in mice, Karikó wanted to go further. She knew there were several obstacles to overcome, but was confident she could overcome them. However, in 1995, she was demoted because no money was coming in to support her work on mRNA. At the time, she had endured a cancer scare, and her husband was stuck in Hungary, sorting out a visa issue. But she hung on at Penn and kept working on the problems until in 2005, she and a Penn colleague, Drew Weissman, discovered that a slight modification to the mRNA solved the problems. They wrote a series of papers which caught the eye of 2 key scientists who would later become founders of BioNTech and Moderna. Ugur Sahin and Özlem Türeci both have Turkish roots but grew up in Germany. Both are physicians who met 30 years ago and pledged to invent a new treatment for cancer. Long before the COVID pandemic broke out, Sahin had read Karikó's papers and was studying mRNA for cancer research. In 2008, Sahin and Türeci found BioNTech to expand their research into messenger RNA. BioNTech is a blended name which stands for Biopharmaceutical New Technologies. Karikó was hired as one of the early BioNTech scientists. On January 25th of 2020, just last year, shortly after Chinese scientists had sequenced the COVID virus, Sahin, working on his home computer, designed the template for 10 possible coronavirus vaccines, one of which is the vaccine currently authorized in both the US and the UK. And despite the facts that BioNTech was short of capital funding, the virus hadn't been diagnosed in Europe, and that it didn't even yet have a name, Sahin immediately realized the huge threat and opportunity and decided to refocus BioNTech on combating the virus. He reorganized his staff into 7-day shifts, asked key workers to cancel their holidays and stop using public transport. His goal was to develop a vaccine in months rather than years, and he named the effort the Lightspeed Project. Last March, BioNTech and Pfizer signed a partnership deal. Morgan Stanley estimated the vaccine would bring Pfizer and BioNTech more than $13 billion in revenue. Sahin has pledged that any proceeds will be reinvested. The fact that this vaccine has been designed, tested, shown to be 95% effective against COVID-19 and manufactured in quantity in less than a year is nothing short of astounding. This is the outline of the development plan or process that BioNTech used to develop their vaccine. Starting with the genetic information from the virus, which had been made public on January 12th of last year, they came up with 20 potential vaccine candidates, performed preclinical testing, narrowed the field down to the top 4. They then tested the candidate vaccine formats on 500 participants in the US and Germany in April and May of last year, and by July had focused on a lead candidate vaccine known as BNT162. They started phase 2 and 3 testing in the US on July 27th, and after phase 3 testing on 43,000 candidates, the final vaccine was approved by the FDA in December. It is known as BNT162b2. The target protein is known as the full-length spike glycoprotein S. Here's a computer-generated image of the spike glycoprotein target. Here's the story of Moderna. Derek Rossi was a Stanford postdoc in stem cell biology when he read Carico's paper in 2005. He saw it as groundbreaking and believes it should earn her a Nobel Prize in chemistry. In 2009, Rossi and one of his postdocs at Harvard Medical School discovered a way to use messenger RNA to reprogram adult cells to act like stem cells. Rossi pitched the idea to Robert Langer, a biomedical engineering professor at MIT, who realized the idea had a huge potential for additional areas of life-saving research. 3 days later, Rossi pitched the idea to Nubar Afeyan at Flagship Ventures, who immediately saw the potential. And within months, Rossi, Langer, Afeyan, and another Harvard physician researcher formed Moderna, a new word combining modern and RNA. Ironically, Rossi is no longer at Moderna, but he had successfully planted the seed idea based on Carico's work that messenger RNA had amazing potential. I don't have details on exactly how that happened at Moderna, but since Moderna's vaccine apparently is also about 95% effective, the company was also able to design, test, and manufacture their vaccine in less than a year. The BioNTech and Moderna vaccines both use messenger RNA technology and were developed and tested in less than a year. Both require 2 vaccinations 3 weeks apart and have been shown in their phase 3 trials to be 95% effective after about 3 to 4 months. The FDA had hoped vaccines would be at least 50% effective, so this is a stunning achievement. Developing vaccines typically takes up to 10 years, but manufacturing and distributing enough vaccines for 330 million people in the US is a daunting task, not to mention demand in the rest of the world. The US introduced Operation Warp Speed last May to fund the manufacture and distribution of 6 promising vaccines, including these 2. Operation Warp Speed was initially funded with $10 billion from the CARES Act passed by Congress on March 27. By October 2020, Operation Warp Speed funding had been increased to $18 billion. The vaccine does not contain the COVID virus, so you can't get COVID from it. It only contains messenger RNA that ribosomes translate into one of the spike proteins on the virus surface. And that concludes my presentation. Thank you very much for paying attention.